Hello everyone, welcome back to my channel. I hope you all are doing well and you are preparing pretty well for the CSIR 2025. So I have previously provided the part 1 of this video. I have extracted different important questions from the standard reference book. So you liked that video so much, so I thought to prepare the second part and different part of it. So I have compiled another important question from the standard reference book, that is Cooper. So all these questions are very important and the chances are very high that this may come in your upcoming exam. So if you like my efforts, you can hit the like button and do subscribe my channel for more such videos. So the first question is, what term is used to describe a gene located within the intron of another gene? So what do we call that gene which is located within the intron of another gene? So that is known as not an overlapping gene, not a pseudo gene, and not a tandem gene. Instead it is known as the nested one. So the nested genes are those genes that are completely located within the introns of other gene. So despite being excised from mRNA, these intron may encode functional RNA or proteins, and when they do, they are called the nested genes. So इस टाइप के जो कॉन्सेप्ट हैं वो CSIR के B सेक्शन में पूछे जा सकते हैं. So you have to prepare it accordingly. So the second question is, what is the approximate percentage of protein coding genes in Drosophila that are considered nested gene? So again, this is a factual question. This is taken from Cooper. So this can be asked in your exam, that what is the approximate percentage of protein coding genes in Drosophila that are considered as nested gene. Nested genes means these are within the introns of another gene. So it is about 5%. So in Drosophila, more than 5% of protein coding genes are nested within introns of larger genes, highlighting a significant genomic arrangement. So you have to remember it that approximately it is 5%. Now next question, in humans, you have to focus on each and every specific term mentioned in the question. Please try to focus on whatever it is asked in the question. So in question it is asked in humans. In the previous one it was asked in Drosophila. So in humans, how common are nested protein coding genes? So in humans it is very rare, it is less than 1%. So whatever paragraph was there in Cooper, as per that paragraph, only about 150 nested protein coding genes have been identified, accounting for only less than 1% of the total whatever gene we are having, making it relatively a rare occurrence. So the right answer is it is rare and it is less than 1%. Now next question. Which of the following non-coding RNAs can be encoded by introns? So it is the small nucleolar RNA and microRNA. So the right answer is option C. Right answer is option C. So both of these are involved in regulation of the gene expression post-transcriptionally. So now back to the next question. Which of the following roles do intron not typically perform? Encode pseudogenes, serve as regulatory sequence, translate into cytoskeletal protein, host nested gene. So the right answer is option C. So this is not the function of, or you can say the role of the introns. So introns are removed from mature mRNA, so they do not translate into protein because we remove them during the splicing process, so they cannot synthesize any kind of protein. However, they may contain pseudogenes, regulatory sequence, or nested genes, so they cannot synthesize any kind of protein because introns are removed from the mature mRNA. So the right answer is option C. Now next question. What step of gene expression can be regulated by intronic sequences? So आप कौन सा चीज आप रेगुलेट कर सकते हैं इंट्रॉनिक सीक्वेंसेस के थ्रू? सो इट इज द ट्रांसक्रिप्शन दैट you can control Intron may contain regulatory sequences that influences transcription, the first step of gene expression. So these regulatory elements help determining cell type specific gene expression. So इंट्रॉन्स में बहुत सारे रेगुलेटरी सीक्वेंसेस होते हैं जो कि ट्रांसक्रिप्शन को इन्फ्लुएंस करता है. सो as we know, transcription is the first step of gene expression, so we can control transcription by the intronic sequences. Now next question: which of the following processes allows a single gene to produce multiple protein isoforms? So we know that alternative splicing is the key. Alternative splicing allows us a single gene to produce multiple protein isoforms. So how it does? It enables different combination of exons to be joined together, like 1 2 3 4. So this is the combination 1 2 3 4. You can do it like this way also, 1 3 2 4, like 1 3 2 4. So likewise you can have different arrangement of exons, different combination of aligning exons in a different pattern. So through that you can produce multiple protein isoforms. So through alternative splicing you can allow a single gene to produce multiple mRNA variants, and hence if multiple mRNA variants are there, different protein isoforms will be made. So this is an important question, so write it down in your notebook. Now next one is, approximately what percentage of human genes are subjected to alternative splicing? So the number is very high. About 90% genes in humans are subject to alternative splicing. So whatever text was mentioned in the Cooper, according to that 90% of our human gene undergo alternative splicing. So it is a very common mechanism in humans to expand the protein diversity from a limited number of genes. So whenever we are having limited number of genes, it is not that we will be having only limited protein diversity, because we are having alternative splicing. So through alternative splicing our mRNA variants will be more and more protein isoforms will be there. So now next question: on average, how many alternatively spliced mRNA are produced from each human gene? So on an average, alternatively spliced mRNA are produced from 6. So how many alternatively spliced mRNA can be produced? So it is around 6. So each human gene yields an average of 6 alternatively spliced mRNA. Out of these, 4 typically encode different protein. So this is an important question. You can include this in your notes. Now next question: what role do intronic sequences play in alternative splicing? So what do the role of intronic sequences in alternative splicing? So as I told you, they contain regulatory sequences which are important for the splicing. So intron regulates splicing by providing sequences that direct where splicing occur, because it guides that from here it has to be spliced. So they provide the, you can say, the sequences that will guide and direct that it has to be done from here. So these regulatory elements ensure exon selection, enabling alternative splicing. So this is the role of intronic sequences in alternative splicing. So now next question: which of the following best explains how the human genome, about 20,000 genes, can produce 80,000 different protein? So just now I told you that it is not so that we have limited amount of gene, so limited amount of protein will be made. No, in humans we are having about 20,000 genes, 20k genes are there approximately, but we are having more than 80k of protein. So what does this mean? Only 20k genes but 80k variants of protein in our body. This is just because of the different mRNA variants which are made due to the alternative splicing technique. So alternative splicing is one of the most important technique which offers the diversity in protein in different organisms. So human genomes only have about 20k protein coding genes, but alternative splicing allow these genes to produce multiple mRNA isoforms, which will result in 80k different proteins. So let me explain the alternative splicing. So this is the figure which was taken from Cooper. So this is the chromosomal arrangement. This is exon 1, exon 2, exon 3, exon 4, 5 and 6 likewise. So you can expect the arrangement like this. The first exon, second exon, the fourth exon, fifth exon and the sixth exon. In here we have not included the third exon. So this can be the first mRNA variant and therefore protein 1, in which we have only 1, 4, 2, 5, 6 exon. So likewise we can have 1, 3, 5, 6. So this will made protein 2 through the mRNA variant 2. Likewise, we can have 1, 3, 4, 5, 6. We can omit the second exon. So through different arrangement of exon, we can have different kind of protein, because we are alternatively splicing the overall chromosomal DNA. We are choosing what we want. So alternative splicing, so this gene illustrated contains 6 exons separated by 5 introns. So alternative splicing allow these exons to be joined in different combination, resulting in the formation of 3 distinct mRNA and protein from the single primary transcript. So it depends on you that you want to take the all exon, and whether the sequence of the occurrence of exon is controlled by you. So through this way we can have the protein variants in our body, despite being having low in overall protein coding genes. Through the alternative splicing we can have different variants. So this is an important question, this is an important topic, you should know about it. So now back to the next question. So the next question is, approximately what fraction of the human genome is made up of protein coding gene including introns? So it is about 33%. So one third. So approximately we are having 33% of protein coding genes, which also includes the introns, out of all the human genome we are having. So it is about one third of the whole human genome. So the rest of it is non-coding sequences with different roles. Now next question. Which of the following statement true about non-coding sequences in eukaryotic genome is true? They are junk DNA with no function. They serve only structural role in chromosome. Many have role in gene regulation and genome evolution. They only occur in prokaryotic genomes. So the right answer is option C. Many have roles in gene regulation as they have regulatory sequences and genome evolution. So non-coding sequences, once called junk DNA, are now known to be involved in gene regulation, chromosome structure, replication and genome evolution, especially in eukaryotes. So previously it was regarded as junk DNA, but later on when the research let us know that they have a very important role in the phenomena of gene regulation, chromosome structure and the replication as well as genomic evolution in eukaryotes. So the right answer is that we are having non-coding sequences for the different roles like in gene regulation and genomic evolution. So this was all for today. I hope you liked it. Don't forget to include all these points in your notes, and don't forget to subscribe the channel because it takes a lot of efforts to compile questions from standard reference book. So if you want me to prepare a particular topic for you, so I will do that. Thank you so much for watching.